Today I will summarize my talk about the HER2DX genomic test. This is a new assay that is clinically available. And this assay is the consequence of 10 years of research in early stage HER2 positive breast cancer, where I think we've learned that we need tools to help us optimize systemic therapies, not only in the neoadjuvant setting, but also in the adjuvant setting in small tumors like stage 1. So HER2DX is an assay that builds upon 10 years of research. It is based on the expression of 27 genes that recapitulate 4 main biological features of HER2 positive breast cancer. On one hand, we have the immune signature, which represents 14 genes among the 27. And then we have a proliferation signature, a luminal signature, and also the expression of HER2 and the HER2 amplicon. And with these 4 biological variables, together with the tumor stage and nodal stage, the HER2DX provides 3 types of information. On one hand, it provides a prognostic score. We call it HER2DX risk score, that was trained using these 6 variables, the 4 biological, the 2 clinical, to predict long-term outcome in early stage breast cancer. And this might be especially useful in stage 1 disease, where we need to make decisions in the clinic, whether to indicate the use of a single taxane and 1 year of trastuzumab versus the standard of care in the past for most patients, which was multi-agent chemo and 1 year of trastuzumab. And also in this stage 1, we must acknowledge the very small tumors, less than 1 centimeter, where the evidence of using trastuzumab or chemo is vague, and therefore having a prognostic tool might help make better decisions regarding what type of systemic therapy in these very small tumors, less than 1 centimeter node negative. Also, the HER2DX, as I said, provides 3 types of information. One is the risk score, as I mentioned. The other one is the PCR score. It's a score that talks about the probability of achieving a pathological complete response after trastuzumab-based neoadjuvant therapy. The score has been validated across many different studies. And basically, this will help. It's mostly in stage 2, stage 3 disease, where knowing the PCR score might help escalate the amount of chemotherapy or de-escalate the amount of chemo and just treat patients with a single taxane, trastuzumab, and pertuzumab. In fact, in the DAPHNE trial, we validated retrospectively and showed that if you are PCR high, which is around 1/3 of the patients, the probability of achieving a pathological complete response with THP 3 months is 92%. So in this particular scenario, stage 2, stage 3, that's where the clinical utility resides. And finally, we provide also the levels, the individual levels of RBB2 mRNA score by itself. They are useful for predicting the true clinical status of HER2 positive versus HER2 negative. But also we have validated RBB2 mRNA as a predictor of response to anti-HER2 therapy in the absence of multi-agent chemotherapy. And this has been retrospective validation in trials such as the PAMELA trial or the PEROLISA. So overall, the HER2DX assay today provides clinically meaningful information in early stage HER2 positive disease that will help and is helping today make better treatment decisions for patients. Thank you very much.